Millions of people with Alzheimer's disease wake up every day slowly losing their memories, their personalities, and their ability to perform simple tasks. To understand why, we have to look inside a brain containing around 86 billion neurons. For decades, researchers tracking the disease have found two primary suspects at the scene. The first is a protein called amyloid-beta, which clumps into sticky plaques outside the cell. The second is tau, which twists into tangles inside the neuron. For decades, this is where the biological trail went cold. Scientists knew these proteins were present, yet they couldn't explain the actual biological mechanism causing the neurons to die. Strangely, some neurons managed to survive despite heavy protein buildup. Meanwhile, other neurons died long before any massive damage was even visible under a microscope. The presence of these toxic clumps alone doesn't explain the death of the mind. A crucial piece of the biological mechanism has been missing. For years, researchers focused on a process called apoptosis. This is the standard way a healthy cell naturally breaks down and dies. Think of it as a carefully planned demolition. The entire cell packs up its contents, breaking down in a highly organized sequence. But the dying brain cells in Alzheimer's patients didn't match those rules. They looked different, meaning they weren't undergoing a standard orderly collapse. If these neurons aren't using the standard self-destruct mechanism, what specific form of cell death is actually pulling the trigger? Identifying the specific biological weapon destroying these cells is the only way researchers can design treatments to finally halt the progression of the disease. The answer requires looking past the outer membrane of the neuron, deep inside its nucleus. This is the cell's control center, housing its vital DNA. A recent study from King's College London and the UK Dementia Research Institute has newly identified a process called karyoptosis. Unlike standard cell death, this mechanism specifically targets and attacks the control center first. Inside the nucleus sits a supportive framework built from a specific protein called lamin B1. You can think of lamin B1 as the internal steel girders of a skyscraper. Their main job is to hold the outer structure up and keep the entire building from collapsing inward. If that physical framework fails, the brain cell's command center is left completely vulnerable to destruction from the inside out. The researchers discovered that when toxic proteins accumulate inside a neuron, they trigger a specific signaling pathway called p38 MAP kinase to become hyperactive. This hyperactive pathway physically attacks and destabilizes the lamin B1 framework until stress fractures begin to form. Without its primary support structure, the nucleus slowly caves in on itself. The DNA inside is severely damaged, and the entire neuron eventually dies. To prove this was actually happening in human brains, researchers examined roughly 3,000 brain cells across 28 different individuals to look for this exact collapse. This chart shows the results. They found features of karyoptosis in 35% of frontal cortex cells from people with Alzheimer's, compared to only 15% in healthy older brains. Science has identified the specific structural mechanism toxic proteins use to execute brain cells. To be clear, this research is still in its early stages. We do not currently have a cure, and there is no approved medicine that can stop karyoptosis in living humans. For many years, scientists primarily focused their efforts on trying to wash away the toxic amyloid and tau proteins from the brain after they had already formed. But understanding how the nucleus physically breaks down offers a new strategic roadmap. What if we could actually armor the cell's control center to withstand the toxic environment? Future treatments could involve a dual therapy approach. While one drug is busy sweeping away the harmful proteins, another could form a protective shield, blocking the specific chain reaction that crushes the nucleus. The medical goal expands from merely cleaning up the biological crime scene to actively protecting the cell before it reaches the point of no return. The implications of this discovery reach beyond Alzheimer's disease. Researchers also found signs of karyoptosis in frontotemporal dementia. And in laboratory testing, models of ALS displayed evidence of this same nucleus-crushing mechanism. This diagram highlights a remarkable possibility. Distinct neurodegenerative diseases might all share this same underlying pathway of cellular collapse. If future testing confirms this, finding a way to stop this one specific structural failure could eventually provide a foundational method for treating multiple devastating brain diseases at once. It proves that asking better scientific questions, focusing intently on the exact mechanism of a cell's death rather than just identifying the protein surrounding it, is how researchers reach future medical breakthroughs. Let us know your thoughts on this discovery below. To follow more evidence-based science documentaries, like, share, and subscribe. Thanks for watching.